Today we'll be demonstrating the procedure of gram staining. This will be demonstrated by Mr. Vitus Silago, and it will be conducted at the KUHAS Microbiology Lab. The purpose of this procedure is to differentiate between gram reactions, cellular morphology, and arrangement of gram-positive and negative organisms from clinical samples and culture isolates. Reagents and media. Gram stain reagent: Cristoviolet, Lugol's iodine, decolorizer, basic carbofucsin, running tap water, normal saline, control organisms. Other materials for the procedure include sterile 1 microliter inoculating loops, frosted end microscope slides, pencil for labeling, source of flame, timer, dropper, staining rack, biohazard bag and gloves, microscope, and immersion bag. Safety considerations: treat all samples as infectious and therefore observe standard universal laboratory precautions. Principle of gram staining. Gram staining depends on the structure and composition of the bacterial cell wall. Gram-positive bacteria have a thicker peptidoglycan layer than gram-negative bacteria. First, the primary stain, Cristoviolet, stains all the cells purple. Addition of iodine, which acts as a mordant, forms cristovalent iodine complexes within the cell wall. These cannot be easily washed out from an intact peptidoglycan layer. Application of 10% acetone in alcohol decolorizes the primary stain from gram-negative organisms. These have a thin peptidoglycan layer and lipopolysaccharides layer that gets dissolved. To observe the decolorized cells, secondary stains like basic carbofucsin or neutral red or safranin is added, hence gram-positive cells will appear blue to purple, while gram-negative cells will appear pink to red. Procedure steps. For this demonstration, we will use bacterial colonies from solid media. Use a pencil to label slides appropriately. Place a drop of sterile normal saline on the slide. Transfer a small portion of colony with a sterile applicator stick or inoculating loop. Mix gently to emulsify. Smear fixation. Air dry slides on a flat surface before fixation. Make sure the slide is well dried before fixation. Heat fixation. Pass the air dried slides 2 or 3 times through a flame. Do not overheat to avoid distortions. A slide warmer set at 60 degrees centigrade may also be used to heat fit several slides at one time. Allow the slide to cool before staining. 95% methanol may also be used to fix direct smears from clinical specimen. Staining. Place a slide to be stained on a staining rack while the smear is upwards. Flood slides with crystal violet reagent and wait for 1 minute. After 1 minute, rinse slides with a gentle stream of running water. Flood the slide again with iodine reagent and wait for 1 minute. After 1 minute, rinse slide with a gentle stream of water. Hold the slide in a slanted position and then apply the colorizer dropwise until normal color is coming out of the smear. Rinse gently with a stream of water. Flood the slide with a counter stain, basic carbofutsine for 1 minute. If you're using neutral lead, then stain for 2 minutes. For our demonstration, I will be using carbofutsine. Rinse the slide with a gentle stream of water. Wipe the back of the smear and place in draining rack for smear to air dry or hot air oven at 50 degrees centigrade. After the slide is completely dried, the next step is examination of the smear. Examine the smear microscopically by first with a 40 times objective to check the stain and the distribution of material. Next step, use 100 times oil immersion objective to look for bacterial gram reaction, bacterial cell morphology, and their allergy. Results interpretation. Gram-positive bacterial cells will appear dark purple or blue colored. Gram-negative bacterial cells will appear pale to dark red. This is the end of our demonstration. Thank you.